I will be leading the next session, which is titled Solutions that Support the Reliability or Integrity of Test Data. I will be discussing this topic together with a demonstration. I would like to break up today's session into two parts. The first part will follow the list of content written here. First, I will describe what specific measures must be in place to ensure data integrity and the required scope of those specific measures. The second part of this session is titled Summary of the Report Set. In this part, I will look at what kind of system and what kind of features are required for data integrity. We have developed a new function incorporated in a new product, the report set, that can meet these requirements in a simple way. I will also be describing how the report set is actually created and using these presentation materials to explain and introduce this method and the main features of the report set. Also today, as well as covering data integrity, I have also brought along software that is actually used in the field. As a demonstration, I will be using the actual software to create a report set. But first, I'm going to talk about data integrity. Data integrity has already been mentioned in many other presentations. Each presentation will have arrived at the question of what must be done to ensure data integrity. Essentially, we need to ensure the reliability of all activities. This topic has already been discussed many times. On the left, I have put up a complete but simplified workflow. The red areas are inputs. If I had to describe them, the inputs are things defined in SOPs, such as sample information used to perform necessary tests and what tests to perform. If these inputs are not correct to begin with, we cannot guarantee the reliability of subsequent test data. It is important that this information is input in the correct and proper way. After the inputs in the red areas are complete, in the blue areas, analytical activities are performed based on the inputs. These are actual analyses and sample measurements carried out according to the inputs. Once samples are measured, it is important to ensure a protected data life cycle of creation, modification, storage, and destruction, and also that the analytical data are protected in a computer system. Correct test data are only achieved once the entire data life cycle, from generation to modification, storage, and destruction, is secured. So the matters below this line can also be secured by a computer system. Such a computer system must, as a matter of course, include the features required in Part 11 and ERES guidance. Shimadzu believes that whatever is under this line can probably be handled by a computer system. But once such a system is introduced, doubt arises about the scope of data integrity requirements in that system. I am assuming everyone here uses a variety of instruments in their laboratories. Some of the most important pieces are probably HPLC, GC, and LCMS systems. Of course, many other types of instruments, such as UV and FTIR systems, pH meters, and analytical balances are also used. Of these, UV and FTIR systems and pH meters are very simple, whereas LIMS, ERP, and MES are very complex, both in terms of systems and software. And as each instrument is different, the validation required for each instrument differs depending on its complexity. Highly complex instruments in particular require careful validation. Meanwhile, relatively simple systems such as UV or FTIR are mostly operated as standalone systems. Of course, when an instrument is isolated and not managed by a server, the status of the instrument is not known, and tests can be repeated on the instrument until the desired result is acquired, all hidden from inspection. The scope of data integrity does not only encompass HPLC and GC systems. Data integrity must be evaluated and ensured for other systems, such as UV and FTIR systems. Considering the scope of data integrity encompasses and affects a variety of instruments, it becomes important to determine what is required of the system that controls such instruments. Shimadzu has a product called LabSolutions, which is a system designed to control Shimadzu's instruments. LabSolutions is available in DB and CS versions. These versions are both able to manage a database of analytical data securely, including all kinds of analytical data, from PDF files to raw data. Since managing data on a database is becoming standard practice, I'm going to look at this topic closely while also introducing some other features of LabSolutions. The first feature is its management of a database of analytical data. Second is the capacity to implement change control for analytical data and methods. In this situation, analytical data refers to the changed parameters, who performed the change, when the change occurred, and why the change occurred. Information that is recorded within the test data whenever a change is made to acquired test data. LabSolutions can create this change control data and manage it securely in a database. Third is the ability of LabSolutions to record an audit trail of operations and errors. These operations and errors are in the form of an operational log that can be used to confirm when an analysis was performed, when the acquired data was entered into the database, which operations were stopped temporarily or underwent any other operation, and that such operations are properly entered into the database. This operational log is also managed securely in the database. In addition, LabSolutions is able to control many different kinds of instruments, not only HPLC and GC systems, and manage the data and raw data from each of these kinds of systems. I mentioned earlier that Shimadzu's LabSolutions is available in DB and CS versions. I am now going to briefly describe each of these products. LabSolutions DB manages analytical data from a single computer. For example, LabSolutions DB is designed for customers with a few instruments, such as 3 or 4 units, to manage these units from a single computer. LabSolutions CS, meanwhile, is designed for customers with more instruments, such as tens or hundreds of pieces. LabSolutions CS is designed to manage the data from all analytical instruments from a single server. The scope of what LabSolutions CS can manage includes HPLC, GC, and LCMS systems, and can include features that allow remote control of these systems from any computer connected to the network. I would like to now give a brief description of what kind of instrument is connected in a LabSolutions CS system. One of the major features of LabSolutions CS is that it manages analytical data from various instruments centrally on a server. For example, placing a LabSolutions server here would allow all the data from analytical instruments in laboratories connected to this server to be stored securely on the server. In this configuration, data from LC and GC systems, and data from FTIR and UV systems and other instruments inside this line are all stored on the LabSolutions server. UV and FTIR systems in particular are being mentioned more and more often in data integrity guidelines. I believe it will become an important issue in data integrity for particular kinds of data to be managed securely in a particular way. I would now like to describe a real-world example of ensuring data integrity in which an instrument was procured that could properly clear Part 11 and FDA guidelines. This example actually took place with Mr. Yamasaki's department from Takeda Pharmaceutical. I will describe just how they went about ensuring data integrity. They started by deciding what information was needed to meet data integrity requirements and decided to operate on a paper basis. In Takeda Pharmaceutical's case, the initial assumption was to have procedures in place allowing a testing manager to confirm for each batch whether or not testing was performed according to established procedures. In this case, it was decided that this would be done operationally on a paper basis. First, they had a printout of a batch table showing required information and information they wanted to acquire. The printed batch table shows actual analysis information, including a variety of data from each individual batch and the conditions used in obtaining the analytical data, which included information such as sample names, sample IDs, and methods used. Second was to print out an operational log pertaining to the data included in the batch table just mentioned. This is the audit trail. They identified information relevant to the operational log themselves and printed it out. Third was to print out information on the methods that appear in the batch table, such as the parameters of the instruments used for acquiring data and analysis parameters. Fourth was to print out a report with the test results. This was an analytical data report of the kind everyone probably prints in the course of normal activities. Sample information, chromatograms, and quantitative results were printed for each batch. A testing manager would then print this information and check it, and if they found no issues, store the printouts. Since this was all to be done on paper, as Mr. Yamasaki mentioned, some aspects were quite difficult. The first major problem was that everything was printed individually, which was very time-consuming. Printing documents really takes up a lot of time, taking away from time that could be spent on analysis work. Plus, as mentioned in the earlier presentation, extra staff are needed. The second major problem is that everything is performed manually. Manual operations bring with them the opportunity for mistakes to arise. The third problem is that you cannot guarantee that data was not edited after being printed. Since this is a paper-based operation, it is possible to mistakenly repeat an analysis after data for the original analysis has already been printed. And the final problem is that storage of these printed materials soon becomes an issue. A large number of tests are performed in the course of daily operations. All four kinds of information from each batch mentioned earlier need to be bound and managed together. Creating a library to manage the printouts in those binders and managing each binder of printouts within that storage space is a difficult task. Furthermore, you would need procedures for entering the library and also to create records of each admittance. This is one example in which data integrity requirements were met. But now I am going to look at an example FDA warning letter that was written in response to a site that only used paper documentation. The FDA inspector's observations included finding ripped-up paper with raw data on it in a disposal area. I think it was mentioned in an earlier presentation, but inspectors will look in trash cans. I will briefly describe what the problem is. Take, for example, a batch that has in total 5 analysis reports. The reports are created as paper documents. Assuming the third test report contains an unacceptable result, but the fourth and fifth reports contain acceptable data, the third report could be redone and the unacceptable report replaced. Another problem could be if, for example, results in the fifth report are ignored and thrown away because they turned out a bit strange. These are the kinds of problems that can arise with paper reports and when working with paper documents. Paper reports can be easily replaced, but there must be a way of ensuring the information on paper reports is consistent with the latest electronic records. With Mr. Yamasaki, we investigated how this problem could be resolved, and as a result of looking for a simple solution to this problem, I would now like to describe the report set, which is a new feature in LabSolutions that provides and evaluates data integrity. First, I would like to give a brief outline of what the report set feature consists of. The basic functions of the LabSolutions report set are to automate creation of the 4 types of information mentioned earlier and basically gather them into a single PDF report. I will now remind you of that information. The first is an analysis register, which contains information related to the batch table. This feature in LabSolutions initially outputs, in the form of a register, a list of information that includes how much data are included in the batch and the sample information mentioned earlier. So the first thing to be output shows at a glance and in list form how much data are included in the batch. The second is an audit trail for the batch data, which is output automatically and collected in a single PDF file. The third type of information is a report of the methods used in the analysis register, such as instrument parameters and analytical parameters, which are output automatically and collected in a single PDF file. The fourth type of information is a report of analysis results related to all data included in the analysis register. All of this information is collected in a single place, with LabSolutions being designed to gather the information as a single PDF file and add the PDF file to a database. I would like to describe in a little more detail what kind of information is output in each of these documents. First is the analysis register. This shows all the data in the report set listed on the first page. As an example, we can understand at a glance of the first page there are 11 sets of data. Listed for each data file are the name of the file, date and time of actual analysis performed, analysis type, and other information, such as whether the data has undergone manual peak integration. It also shows basic information for each analysis, such as the tray number and vial number. Not only that, this part also shows the ID of the method used for each analysis. Numbers can be shown here to allow unique identification of the method used to obtain each set of analytical data. For example, if you want to know which method was used for this analysis, this number 10 can be used to uniquely identify the analysis method used. This number is also the unique number in that system. This feature has been added to allow easy identification of analysis methods. Next, the report set audit trail is extracted automatically. Investigative information is extracted automatically from data files used in the batch, then displayed and registered in the audit trail. Automatically included in the audit trail is information such as the operational log of analysis, which includes when the batch was analyzed and whether a temporary stop was applied. Also added to the audit trail is the operational log of data analysis, which includes when data analysis was performed and what data was analyzed. This log also contains instrument information, such as error information that allows confirmation of whether an error occurred during analysis, and if so, whether it posed a problem. The information in this log covers the duration of the analysis, from operator login to operator logout. This information is gathered automatically and output as the audit trail log. Third is a method report that is output automatically as part of the report set. The method report shows all analytical conditions, including detailed information on instrument settings, and basic information about the analysis methods such as pressure levels during analysis and duration of analysis. The method report can also output conditions used in data analysis, such as the analyzed parameters, slopes, and width. All this information is included in the method report. The method report also, of course, can include the method ID. So checking the method ID in the method report provides a simple means of identifying which method on the system was used. Finally, a data report is also output as part of the report set. This report includes the kind of information everyone views and uses in the normal course of activities. It includes basic information about the data file, but also analysis results, chromatograms, and quantitative results. One of the basic pieces of information included in the data file that is also output in the data report is whether the system's audit trail was turned on or off. Of course, information such as the analysis method used to obtain data and what method number was used can also be output in this report. The report is designed to allow unique identification of which method was used based on the method number. These 4 types of information, referred to in this presentation as an analysis register, audit trail, method report, and analysis result report, are generated automatically and output as a single PDF file which can then be added to the database. These are the basic features of the report set. Now I will discuss the actual information that appears in a report set and explain the workflow of creating a report set. LabSolutions is designed so the report set can be created in just 3 steps. The first step is to launch Data Manager. Data Manager is launched from the main window of LabSolutions. Data Manager is an application that allows the user to view all data in the database. Launching this application is the first step. The second step is to select the batch file or data files used to create the report set. I believe some manage files as a batch. In that case, you select a batch file. If you manage by data sets and not by batch, select data files to include in the report set. The first example will be for selecting a batch file, so the batch file is selected here. The third step is to create the report set from the selected batch file, which is done in a single click. The created report set can then be checked on screen. Finally, once the report set has been checked on screen, assuming there are no problems, the report set is saved and the creation process is complete. All this takes just 3 steps. The report set can be created in just a few minutes, which reduces workload substantially. I would like to describe this process a little more, but this time using the actual windows shown in LabSolutions. You launch Data Manager from the LabSolutions main window. Data Manager is shown in the main window. When you launch Data Manager, the Data List window appears. When this data list appears, Data Manager has launched. Next, you select which batch file to use in the report set. The list of batch files is shown here at the bottom on the left side. You select a batch file from which to create the report set and right-click on the batch file, which brings up the Create Report Set menu. Clicking this menu will display the report set on screen. At this point, you check the report set. If no specific problems were found while checking the report set on screen, you select the Save button at the bottom. Selecting this Save button adds the report set PDF file to the database, and so completes report set creation. If you wish to print out the report set at the same time as creating it, here is the Print checkbox. The report set can be printed at this point if needed. These are the basic features of report set creation, explained in the form of the creation workflow. Next, I would like to describe a number of features in LabSolutions designed to support the report set. I just talked about LabSolutions being able to output a report set and method report. Method reports can be output for individual batches. Method reports used for data acquisition and analysis can be output, and a maximum of 3 types of method reports can be output from the batch table configuration window. For example, the results file from Data Manager is created for the settings specified here, and the resulting PDF file is added to the database. LabSolutions is designed to allow the user to check the actual method report at this point. This is one way of dealing with method reports. Another feature of LabSolutions is to prevent falsification of reports by automatically locking all data files related to a prepared report set. Once locked, this data cannot be edited. This prevents reanalysis of data or unintentional changes to data. Also, if a report set is printed, you may not know whether that printed copy of the report set is the latest version. Using the data lock feature prevents reanalysis of data after report set creation, ensuring that report set is always the latest version. When data from the report set is locked, it cannot be reanalyzed unless the lock is removed by a supervisor, so only people with certain permissions have the authority to remove the lock. Another feature of the report set is the ability to link paper and electronic records. This is based on all generated report sets being managed with an ID. This ID uses unique ID numbers in the LabSolutions system. The ID can also be displayed in the paper printouts and the record of the report set being output on paper. For example, in this window, the ID of this report is number 021. If this number is used, then the associated electronic records are also given the number 021. This makes it easy to search for and identify electronic records from the paper records. The fourth feature allows for convenient implementation of a paperless system for customers who want to operate without paper. It allows for a signature to be attached to a report set once it has been created. Attaching a signature to a report set also attaches that signature to the data files used to generate the report set. This feature means that when operating on a paperless basis that requires you to attach a signature to each individual data file, there is no need to open and check each data file. You can just view the report set and attach a signature collectively after checking to ensure there are no problems. This feature not only allows collective review and approval, but also allows the collective removal of signatures from the report set, and by extension, all associated data files. The fifth report set feature prevents a user from skipping the confirmation step. With paper-based operations, I imagine you would go through checking certain boxes to show that an item was actually reviewed. For example, checking and confirming a chromatography report has been created, and checking the results report. I imagine many customers operate in this way. For these customers, when they migrate from paper-based to digital-based operations, it is difficult to maintain the record of whether each one of those items has been checked. As a result, items may be missed. In order to prevent this, LabSolutions is designed to allow the user to designate any point as a check item, to leave an associated record of whether the item has been checked, such as when the chromatogram absolutely must be checked. These check items can be added anywhere, allowing the user to freely customize the most important parts of the analysis based on their operations. These check items can be added at multiple sites, and the process configured so a problem with a particular item will produce a rejected result. For example, say some unacceptable data is added and I try to save it. The list of results shows rejected. This allows the user to see at a glance whether data is acceptable and immediately decide whether reanalysis is required. Also, say for instance you configure 5 check items, but the operator mistakenly only checks 4 items and only enters an accepted/rejected result for those 4 items. When that operator tries to complete the check by clicking Save, an error will appear. This feature is very useful for preventing any check items from being missed. I have now described the basic features of the report set and a number of features designed to support the report set. I believe a report set improves the efficiency of both data integrity-related operations and assessing data integrity requirements. I will now give 4 distinguishing advantages of the report set. First, the accuracy of batch-related reports is ensured. The 4 types of information are gathered automatically when a report set is created. Both report set creation and information collection occur automatically, which prevents operational errors and the incorporation of arbitrary information into reports. Second is that report sets are created automatically from the relevant batch file in a single click. This saves a substantial amount of time. Third is that report sets are managed securely in a database in PDF format, so the content of these PDF files cannot be falsified. The report set is added to the database in PDF format. Also, once the report set has been printed, any further printouts of the report set will be identical to the original printout. Fourth is the ability to ensure that the report set contains the latest analysis results. LabSolutions allows all data files to be locked at the report set creation step. Using this feature prevents the reanalysis of data files once a report set is created, which ensures the report set information is always up to date. Those are the 4 distinguishing features that I will cover later in a demonstration of the LabSolutions software. Finally, I would like to briefly describe the workflow once report set has been introduced. This is a simple example in which roles are divided between an operator and testing manager. First, the operator performs batch analysis. In this case, if there is a problem with the analysis, the operator can decide to perform reanalysis. And if the operator finds no problems throughout the entire batch, they report to the testing manager that all testing is complete. The operator then creates a report set that is checked by the testing manager. If no problems are found during this check, the report set is approved. This workflow is probably as simple as I have briefly described now. I will now go through some recommended check items. LabSolutions is designed to allow the analysis register to be used as a reference when checking the report set to find out if, for example, the prescribed batch or the initially well-defined batch is being executed properly. Second is that everything that allows a determination of whether reanalysis was appropriate is included in the data report and method report. These reports also contain information that shows a user, when viewing the report set, whether the reanalysis was implemented appropriately. An audit trail is also generated. Whether any dubious actions such as temporary stops or interruptions are present in the audit trail can also be determined just by looking at the report set. The final recommended check item is probably something that everyone does in the normal course of work. Whether information such as the chromatography report is correct or not can be confirmed just by checking the report set. This is just a brief example. Next, I would like to describe the creation of an actual report set. First, double-click the LabSolutions icon and log in. Once logged in, the LabSolutions main window appears. I have obtained some actual analytical data for the purpose of this demonstration. In the main window is an icon labeled Data Manager. When you double-click the Data Manager icon, the data in the database appears in a list. For example, this is the data obtained for this demonstration in a list. The fact that this information appears shows the data are already present in the software. To create the report set, first you select a batch data set. When selecting a batch data set, you decide which batch you want to create the report set for. For example, here you can search for user names. You can easily search by role, such as System Administrator or Operator, and find a batch based on that criterion. For example, to create a report set for this batch file, select the batch file. Right-click, and the Create Report Set menu appears. The data displayed on the right here shows information about all the data included in the batch file. For example, if we examine this window, we can see this batch file contains 5 data files. I'm going to select the Create Report Set menu. When you select the Create Report Set menu, the 4 different types of information mentioned earlier, the Analysis Register, Audit Trail, Method Information, and Results Report, are assembled automatically. The information assembled is also displayed in this window. This window is designed to let you first check the Analysis Register at this point. When you first look at the Analysis Register, it shows the 5 pieces of data mentioned earlier. From this view, you can confirm the data contained in this batch consists of these 5 pieces of data. You can check in the window, as mentioned earlier, to determine whether peak integration was performed, which tray was used, and on the second page, the vial number, sample type, and method information. The software is designed to let you configure which of these items is displayed or hidden, and whether other information is also displayed. The quantity of information shown can be increased or reduced as desired. Once these items are checked, next to appear is the Data Acquisition Report, with the operational log and audit trail. Check this window to determine if there is any problem with the actual acquired data. The information shown at this point includes all operational information for the analysis duration, from operator login to logout. This part is designed to allow you to check whether there are any problems. Next is a report of instrument parameters. A list of information about the method used for analysis appears. This part is designed to allow the user to check which conditions were used to perform the analysis. This part also displays information on the parameters used for data analysis in terms of data processing parameters. And finally, I have shown calibration curve information as an example. Data from each of 5 analyses have been assembled in the form of a final chromatography report. The results of these 5 analyses are also included in the single PDF file. LabSolutions is designed to allow you to check whether there is any problem in this information. Let's look at this list. LabSolutions is designed to allow you to determine what kind of data is contained in the analysis register by viewing the bookmark. After checking that the analysis register, logs, method report, and 5 data reports are included, you finally press the Save button, and the report set is registered here in a PDF format. At the bottom appears a list of the data contained in the report set. This section allows the user to easily see what data are included in the report set. Double-click here to again check the already confirmed data and report set, in the same way as they were checked before. With just a single click, you can register a report set with the 4 types of information mentioned earlier. It also shows whether the files included in the report set are locked. A little earlier, we created the report set from these 5 data files. This section shows whether the data files for that data have been locked. locked data cannot be reanalyzed, which ensures the report set created here is always the latest version. And if you intend to use the report set as a digital document, an electronic signature can also be attached in this window. For example, select the report set that was just created, right-click, and select the signature. After selecting a signature to attach to the report set, the PDF of the recently created report set appears in the window. At this point, you can make a final check for problems in the report set. The report set confirmation screens can be used on monitors in landscape orientation, and doing so makes them much easier to attach a signature to the report set. If there are no problems with the report set at this stage, a signature is attached, comments inserted, and on clicking the OK button, a password entry window is displayed. This is in order to prevent forgery. When a signature is attached to the report set becomes digitally signed. Furthermore, a signature is also attached to the PDF file associated with the report set. The system is designed in this way to remove the need to open and confirm each individual data file before signature attachment, which saves on labor. You can also operate on a fully paperless basis. Paperless operations are performed a little differently than paper-based operations, but when viewing digitally, LabSolutions has a feature that confirms whether all items were actually checked or not. First, you select which data you want to check and right-click. If you select the Confirm Result File window at this point, a window is displayed allowing you to confirm the content of the PDF file. In this window, once you have checked the data file for problems, for example, you then add to the PDF file whether to accept, reject, or if no problems were found. For example, if no problem was found with the data file, it would be accepted. However, if you require chromatograms to also be evaluated as accepted/rejected, items can be configured to require entry of this evaluation. Looking here, if there was no problem with the chromatograms, you would select Accept, and if there was also no problem with the peak table, you would select Accept. Now, assuming that all items have been viewed, we press the Save button. If an evaluation, accepted, rejected, etc., has not been entered for an item, a message will display showing which items have not been properly checked. This prevents users from missing or overlooking items. Since we will also need to check this in the audit trail, clicking Accept here will also leave a record in the data file that shows this item has been accepted. Using these features allows for paperless operation while ensuring that electronic records are properly checked in the same way as paper records. This has been my demonstration of the basic features and support features of the report set. Thank you for your attention.